Hello everyone, welcome to today's lecture. Today we'll learn about recombinant protein production. This process is a core technique in molecular biology and biotechnology, especially useful for producing specific proteins in large amount for research or medical use. In this chapter, we will cover three main parts. First, we'll go over what recombinant protein production is and why it's important. Then, we'll learn the step-by-step procedure. Finally, we'll look at how to read the results, focusing on protein purification and visualization. Let's start with a simple overview. On day 1, we begin with a seed culture of E. coli that has been transformed to produce our target protein. On day 2, we scale up the culture and induce protein expression using IPTG. And on day 3, we lyse the cells, purify the protein using a nickel NTA column, and analyze it using SDS page. To start the experiment, we prepare a small-scale culture. We take 4 ml of LB medium in a 15 ml tube, add 4 microliter of ampicillin, and inoculate it with 100 microliter of our transformed E. coli strain. This culture is grown overnight at 37 degrees Celsius in a shaking incubator. The shaking helps the cells stay aerated. On day 2, we scale up our culture by transferring 3 ml of the overnight seed culture into 300 ml of LB medium with 300 microliter of ampicillin. We incubate this larger culture at 37 degrees Celsius. When the OD600 reaches 0.5 or higher, we add IPTG to induce protein expression. The final IPTG concentration should be between 0.5 to 1 millimole. We'll let culture grow for at least 4 more hours after induction, then centrifuge to collect the cell pellet. On day 3, we start by resuspending the pellet into 5 ml of lysis buffer. We then syndicate the suspension for 30 minutes with short pulses, 10 seconds on, 10 seconds off at 30% power. After syndication, we centrifuge at 10,000 G for 45 minutes at 4 degrees Celsius. The clear supernatant contains our protein and will be used for purification and SDS page. Next, we purify the protein using nickel NTA affinity chromatography. First, we load the slurry into the column and wash it with a washing buffer. Then, we apply the supernatant so that his-tag proteins can bind to the column. After washing again to remove unbound proteins, we elute our target protein using an elution buffer. To analyze the protein, we use SDS page. We prepare the gel and fix it into the electrophoresis chamber with running buffer. Our protein sample is mixed with loading dye and heated to 100 degrees Celsius to denature the proteins. We load the samples into the gel wells and run electrophoresis. First, at 80 volts until the stacking gel, then at 110 volts during the running gel. After electrophoresis, we stain the gel with Kumasi Brilliant Blue for 30 minutes and then destain it to visualize the protein bands. The final step is analyzing the gel. You should see bands representing the purified recombinant protein. The intensity and size of the band tell us how much protein was expressed and whether it's the correct size. This concludes our overview of recombinant protein production using E. coli. By understanding and practicing each step, from culture to SDS page, you will gain valuable skills in protein expression and analysis. Thank you for your attention.